record this for the final. You have your final study guide. Use it. Today we're going to go over one last thing. This week we're going to go over photosynthesis and cellular respiration. But to just bullet point it real quick, cell organelles, and function. You have to know what they are. What does each organelle do? Where are they? Cell types. You have to know which cell types are which and which has which organelles. You have three major ones, two big ones, prokaryote, and eukaryote. Prokaryote is a bacteria. Eukaryote, you have animal, plant, fungi, and protist. So you have to know each one of these has different structures. This one, no nucleus. Again, I'm just going to make a real quick summary by doing a Venn diagram with 4 circles. That looks a little complicated. What do you think? What if I do different colors? And I say, this is animal. Well, an animal cell has a nucleus. Golgi body, endoplasmic reticulum, rough and smooth, RER and SER, vacuoles, cell membrane, all that, right? All the cells, all the eukaryotic cells have those. Animal cells, mitochondria, let's not forget the mitochondria. All the animal cell, all the cell types have mitochondria. All the eukaryotes have mitochondria. What you would do best is to try to make this chart on your own. It would be the best thing you can do for yourself, is to make this chart on your own. Can you do this on your own? Do you know? You don't have to make a Venn diagram. You can make something simpler. But you should be able to identify what organelles go with which cell type. So if we would do a plant cell, plants have all of those. They all share all those in common. They share with fungi and some protists fungi and some protists, plants share cell wall. Plants have chloroplast, though, that fungi and protists, well, do some protists have chloroplast? I'd have to look that up. Protists are strange little organisms. But for sure, fungi don't have chloroplast. So now, if we go to fungi, we're talking about yeasts and mushrooms, right? Molds. They have cell walls as well. By the way, their plant cell walls are made of cellulose. And they have something else that other cell types don't have, is a central vacuole. Now, fungi have cell walls, but they don't have chloroplast. So they eat. They have all these other organelles, but they don't make their own food like plants do. They eat. They eat dead things. They're detrivores. Does that make sense? So, yes or no? Does that make sense? Okay. And then the last is protists. They're all over the place. Protists are all over the place. They have cell walls. They have both protists. I'm thinking. Yeah, I think they have a contractile vacuole, and they don't share it with plants. I showed you in an amoeba. You remember that? They just kept pumping water out of the cell. All right. I think if you know that, you'll be able to answer a lot of the complicated questions. It helps. I think it simplifies a little bit with extra color. But you see that most of the organelles, most of the cells share it. Now, to make sure you're clear that a virus is not a cell. Hence, most people, probably the majority of scientists, don't consider them alive, though it's an argument. And it's a bare majority. There's a lot of scientists out there that argue viruses are alive. They're not. They want people to accept them as the exception to the cell theory, because what cell theory say? What does cell theory say? Guys, if you're not working, you're not gonna. Oh my God. The nightmare. The cell theory. What is it? What is cell theory? It's basic. It's right out of the chapter reading, mentioned a bunch of times in class. What's cell theory? Stop trying to get out of work. Cell theory. Come on. That's it. The basic unit of life is a cell. Basic unit of life is a cell. That's right. Now, a virus, continue playing and trying to find ways out of getting work done, but it's not going to help you in the end. No offense, it's just not. So the virus is not a cell. A virus like COVID or like any other virus is made of some kind of protein coat. It's made of a protein. Have all kinds of different shapes. They're very tiny compared to other cells. They're very small. Inside, they have either DNA or they have RNA, single-stranded RNA or double-stranded DNA. Somebody asked, do we need to know DNA? Yes, of course you need to know what DNA is. Of course, we covered it, didn't we? So we covered it. You should know what DNA is. So, you know, at this point, at this juncture, you should know DNA is... Why is this doing this? We know that this DNA, you should know at this point that it's made of A that binds to T, G to C. There's a little bit of DNA inside a virus. The virus just gets taken up by the cell and copies are made, and that kills the cell generally or makes someone sick. And then this is RNA. And in RNA, you have A matches with U and G with C. And RNA has only it's a single strand. And you know that RNA is made of ribose, not deoxyribose, and that kind of thing. Are we all good with this so far? All right, so it's a good review. We're done with that. So what else should you know for a test? You should know how things transport across the membrane. What passes the membrane usually and what doesn't? What is the cell normally, without help, able or permeable to? What does that mean? That means, what is the cell allowed to go through or not? And that means the cell membrane. The cell membrane is structural. It's functional. It allows for both, it gives the cell some structure, also allows it to have some, it controls what goes in and out of the cell. So this is a very important structure. That's why we spend so much time with it. 2 fatty acids, a phosphate group, bilayer, not a single layer, not a micelle, but a bilayer, a fluid mosaic model of cell membrane. What normally goes through and what doesn't? Well, at this point, you should know sodium ion, any charged particle. I don't care if it's sodium ion or if it's phosphate with a PO4 negative. It doesn't matter. If it's negative, small or big, it needs help. It does not pass through easily, not permeable. It needs some kind of helper. At this point, you should know that water passes through somewhat, a little permeable to water, because water's so small, some of it gets through, very, very little, but does go in and out of the cell membrane. That's not new to anyone here, I hope. I hope that everyone here recognizes that sugar and proteins don't get through. They need help. None of these get through without help. What gets through? Anybody know what gets through? What is it that gets through the cell membrane easily? Small and nonpolar molecules. What are they? Oxygen gas, CO2 gas. They go through easily in both directions. I will complete. All right. So that's what gets through without any help. So how do you get stuff? How do you get these things through with help? What is help? Yeah. Some kind of transport protein. So again, looking at the phospholipid bilayer, looking at the fact that you have phosphate heads and you have these hydrophobic fatty acids. You should know this molecule called cholesterol. You see it all over the place. It's a ring structure, has multiple rings. I'm just sketching out a ring structure. It sits in the membrane. I did mention it before, but I didn't stress it, but now I am stressing it. So you should know cholesterol. So cholesterol is a lipid. All right. It sits in the membranes. It increases fluidity. It keeps the membrane fluid, increases fluidity. So it stops the cells from freezing. In the winter, you have more cholesterol in your membranes than you do in the summer. So it helps stabilize. In general, cholesterol stabilizes the membrane. Remember, it's always moving. And obviously, when it's hotter, it's going to be more fluid. You don't want the membrane to be too fluid. Then the other thing that you have, as someone mentioned a minute ago, is you have these things called transport proteins. There's several different kinds that we've talked about in class. The simplest of them being a channel protein that just allows something like sodium ion through going from high to low. Correct? That's the simplest of the proteins. There are other kinds of transport proteins we've talked about. There are some that require receptors and signals. They would have a channel as well, but instead of it just being an open channel, it has some type of receptor waiting for a signal. When that receptor binds to the signal, hydrogen bonding it's a signal molecule binds, hydrogen bonding changes this, and what that'll do is it will open the membrane, open the channel. So when that signal binds, starts off, the signal sent from somewhere else. What kind of things are signals? Hormones, neurotransmitters, right? So some kind of signal gets sent, and when that binds, it changes the hydrogen bonds change, and when the hydrogen bonds change, it changes its whole shape. When that shape changes, this opens up. Because the signal molecule attached to the receptor changes its shape. the door is now open. As long as that signal molecule, or what we call a ligand, right? Do you remember that word? Ligand. Once the ligand is bound to the receptor, the door opens and you can get whatever it is that's getting in, comes in. Again, moving from high to low. That's receptor mediated. We also talked about voltage channels. Do you remember that with the neuron? But you don't have to worry too much about those. The whole point is something opens a channel and things move from high to low. We call this, there's a word for this you should know because you're going to see it and probably see it on the test. Facilitated diffusion. Facilitated means what? To facilitate means to help. If I facilitate your education, I'm helping your education. If I facilitate your removal, I'm helping you being removed. So, help. And diffusion means what? High to low. So it's moving, helping things go from high to low, and here you're talking mostly proteins. Then there's simple diffusion. We talked about that, simple diffusion. Where do you see simple diffusion? That's oxygen and carbon dioxide. It's just going from high to low. No help necessary. Water does that too. When water diffuses, what do we call water diffusing? Osmosis. So all of these are examples of what we call passive transport. Meaning no extra effort necessary, no extra energy required. No extra energy required. This just happens, goes from high to low. All these are examples of going from high to low. High to low, high to low, high to low. There is no extra effort necessary. However, there are examples where you need to go the opposite direction. And we covered that. We talked about it. It was quick. I want to make sure I go over it again with you, because it's that important. I think it's important that we talk about it. There is something called active transport. We spent a lot of time talking about a battery. We talked about a cell membrane can act as a battery. We said a neuron does that. That's right. Uses ATP is correct. Excellent. It's going to use ATP, and in general, there's two things that tell you it's active transport. If you remember this, it probably will get you through every question. Number one, it uses energy. Usually ATP in a cell. And two, it goes from low to high. And the big always the example they use in every book is when you're going from high to low, no extra energy is required. But when you're going from low to high, you have to push that thing uphill, correct? So that's the example in the book. That's the example in every book. That's really the idea that you need extra energy to get it done. If you have this membrane and it's in a neuron, is it time to go? Is that why people are moving? Okay, good. So in a neuron, you have one side's more positive than the other. One side's more positive than the other. And you have these voltage-gated channels. Remember, we talked about them. There are ligand-gated channels. I just drew one earlier. And you have these channels that are voltage-gated. Depending, if you can make it more negative, this will pop open. But you have to change the volts, right? You have to change the ions. And that requires a ligand-gated channel neurotransmitter, allows ions to flow, which starts the domino effect, where you start to get a discharge of the battery. And what's a battery? The cell is a battery, because this side is more positive and this side's more negative. We call that a battery. Why is it more positive? Because there's more positive ions on this side. Maybe there's a ton of them over here and there's only one over here. When you discharge the membrane a little bit, this opens up, and what's going to happen? The ions are going to flow, correct? And as the ions flow, they go down into the membrane. That discharge, that shows up on a screen. You've seen this screen 100 times on TV shows, 1,000,000 times. It goes bleep, 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 correct? Right? And once it stops, it goes correct? So these are discharges of the membrane. Repolarization, these are depolarizing, meaning they're becoming less polar. They're discharging the battery just like your cell phone. Well, what has to happen to recharge the battery? So here, let me follow along, just like on the TV shows. It discharges, then you have to recharge the battery. Discharges, recharge the battery. So this is the discharging when the positives flow through from high to low. That's fine. This part's passive. It's ligand gated and voltage gated. Fine. But what happens? How are you gonna make it polarized again? What do you have to do? Nobody? Just guess. Nobody knows. It's not something any of you know. What are you gonna have to do? So now you got a few, let me draw it out for you. You have a few positives on this side. You have a few positives. They're equal now. You've depolarized it. Maybe even you got more positives down here than you hyperpolarized it, so you actually made it more positive inside than you really want. You have to have a helper. So here's your helper. Let me redraw this. You have to have a helper. When the helper is closed the way it is now, nothing's happening. What's this thing going to do? What do you have to do to make it polarize again? I'll say the question again. The inside right now is slightly more positive, correct? And the outside is a little more negative. We have to reverse that. How are we going to reverse it? Okay, so do I have an answer? All right, go. That's right. We're going to use ATP that you made during cellular respiration, and we're going to move potassium ions. Potassium ions, you see them here. You're going to exchange potassium ions on the outside. You're going to bring them in. And you're going to get 3 sodiums out. Now, each sodium is a +1. Each potassium is a +1. So 2 going in, 3 going out. what are you going to end up with? What's going to happen to the membrane? It's going to become more positive on the outside. And you're going to use ATP to do it. This is called the pump. Sodium-potassium exchange pump. You'll find this example and this explanation on this website. It's a nursing website. I just want you to see how important this is. This is in nursing and dentistry and medical school, any biologist, anybody intent on understanding any living cell needs to understand the sodium-potassium exchange pump. This explains muscle contractions and thoughts. So be ready to talk about this when we get back tomorrow.